Hi, this is Miss Litton, and this is lunchtime. Where are the rest of you? We're doing an honors bio review. Say hi. Hi. All right, and we're doing chapter 4, all about cell membranes. So, first primary thing you need to know is what are the structures that make up the cell membrane and what are the jobs of each. Okay? What are the structures that make up the cell membrane and what are the jobs of each? You are sitting next to somebody right now. Talk about what structures do you know make up the cell membrane. Just identify them. Verbalize it. Go. Oh my gosh. All right. What am I drawing right now? Phospholipids. Good, good. Phospholipids. And phospholipids, their job, phospholipids, they are a, if I picked one word, they are a, starts with a B. I'm thinking of a word, barrier. Okay, tell me something about this portion of the phospholipid. Is it hydrophilic or hydrophobic? Hydrophilic. Water-loving. It's polar. Yes? So these sections are either facing the interior of the cell or the watery medium that it's sitting in. But this portion right here is what? Hydrophobic. And that's the part that truly acts like a barrier because things can't pass through that easily. All right, what am I drawing right now, do you think? Yeah, that's perfect. Yeah. I'm doing a channel protein right there. Or any kind of protein. Good. So this right here, that is a protein. Now, let's review all the things we know a protein can do. It can be a channel. Good. Okay, I'm writing that one down. Channel. It can be a carrier. So good. Review these with your bio buddy right now before I ask you out loud. Do them all. And I'll ask you.
Channel. Carrier. I live on nothing but pure.
Oh, I'm seeing lots of good things.
Wisdom.
We do.
No, wait. Receptor.
Okay, ready? So let's do them together. Ready? You can be a channel, carrier, cell recognition, receptor, enzymatic, and junctions. Yes?
What's the difference when, where's the one where you like, whatever, is that?
I'm going to hit that.
Okay.
That's how you cross it through the phospholipid, right? Okay. So we have channel, carrier. What else do you want to tell me? Cell recognition, cognition. What else?
Enzymatic.
Receptor. What else? Enzymatic. And what else? Junctions. Are we good on that? Okay. What are the other parts of a cell membrane? Glycoproteins. Good. So you could have, let me draw another little protein here. Okay. You could have this. What is that made out of? Carbohydrates. Glycoprotein. Okay. Or it could be attached to a lipid and be a what? Glycolipid. Okay. And that can help with this right here. Cell recognition, right? Part of the glycocalyx. We talked about MHCs. Good. What else do I have? I'm not seeing it yet in here. Animal cells have it, not plant cells. Animal cells have it.
Extracellular matrix.
Okay, you can have an extracellular matrix, and I would say this is probably part of that ECM. I agree with you on that. I'm looking for something else. You worry about having too much of it. Cholesterol, right? Okay, cholesterol. And if you remember, we talked about that having to assist with the fluidity, right? Cholesterol will help keep the cell membrane together if it is too hot, and prevent it from freezing if it's too what? Cold. Okay? Do you want me to do something with that? You want me to save that and Instagram it out or something?
Yeah,
Yeah.
that'd be nice.
I know. Okay, now, what are the ways things can cross a cell membrane? Here, I'll take a picture of this and then I'm going to mess it up. Picture. Okay, good. Hopefully I remember to send that out. Okay, what are the ways you can cross a cell membrane? Okay, so you can go through the phospholipid, okay? But if you go through the phospholipid, remember we did this, you can't be what? Large or charged, right? They can't go through the phospholipids. Okay, what's the second way you can go? Through the proteins. Okay? So there you're either using a channel or a what? Carrier. Or it can be what? Whole membrane. Remember? Endocytosis and exocytosis. Okay? So now let's talk about energy. What are the two big categories? We know where you can get across. Through the phospholipids, but you can't be large or charged. You could use a channel or a carrier. Okay? Or the whole membrane could do endo and exocytosis. Just a minute. I don't like the way that looks. And I'm in the middle of a review session. Is everybody okay? Okay. All right. Let's talk about energy relating to, here, ways to cross. Oh my gosh. I'll change it. Okay, ways to cross. We said through the what? Through the phospholipids. Phospholipids. What's the other way? Proteins. What's the third way? Carrier. Whole membrane. Naughty children, I don't know. Okay, whole membrane. Okay, so you can't do through the phospholipids if you are large or charged, okay? Proteins, you could use channels or carriers. Okay, and whole membrane we can do endocytosis or what? Exocytosis. What are the three types of endocytosis? Penocytosis, which is what? The drinking, cell drinking. What was the other one? Phagocytosis, which is what? Cell eating. And there was receptor mediated. Yeah. Okay? And endo, it's going in, and exo, it's going out. Okay, now let's talk energy and all of this. Okay? If you go through the phospholipids, or you have a channel, that's going to be what? Passive. Right? No energy is needed. Because you're going with your concentration gradient. That could be an active diffusion or osmosis. Does that make sense to you? Okay? But if I go against the gradient, okay, then I'm going to, like, if I'm using a carrier and going against, or if I'm using a whole membrane, that's not going to be passive. That's going to be what? Active. Okay, that's going to be active transport if I go against the concentration gradient. You all right so far? Okay, now I want to talk about something else. When we talk about going through the phospholipids and we specifically talk about water, we talk about there being a membrane and a solution on either side of that membrane. You have some sort of membrane barrier. What do you call the side, okay, that has more stuff on it? You would call that what? The the one that has hypertonic, and this side is hypotonic. And water must what? Flow from the hypo into the hypertonic medium. Okay? And when that happens, when water flows like that, what do we call that? Osmosis. And when it's same on either side, it's what? Isotonic. Okay, now look at me. I can tell by the way you're answering my questions you haven't reviewed this yet, right? And this is your first chance at looking at it since we talked about it in class. Is that an accurate?
Yes.
Okay. All right. Yeah, okay, just so we're clear. Okay, so do you remember talking about this?
Yes.
Okay. So it's going to be important for you, okay, to maybe go back and watch some ed puzzles, okay? Read your notes over and look over this, because what I'm talking about, you should be like, boom, 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 I know what this is. I'm sure you had a busy week, and you haven't had a chance yet to look at it, but I'm just telling you that it would make the reviews more beneficial if you've looked at these things, so when I ask you questions, you're not going, okay? Yes?
So in our notes, it says for hypotonic solutions, it's less solute and more solvent.
Mm-hmm.
But how does that work if there's more?
Okay, so you have two things you're dealing with. The water amount, okay, and the solute. So here, I've got water going across here. This is 100% solvent, just water. This is water with, let's say, some sugar in it. That would be the solute. So the hyperness, the lot, refers to all these other things that are dissolved in the water. Okay? And so water is going to go from where it's more concentrated to where it's less concentrated. So water must flow from the hypo into the hypertonic. Okay? And that process going through the phospholipids of water, that is osmosis. Okay? And if it's same where the same amount is moving in as moving out, that's isotonic.
And does it have to be water or it can be anything?
No, it could be another solvent, but since cells are mostly water, you're most concerned about water movement. Okay? And in fact, eggs, when we did our lab with our eggs, right? All of your eggs gained mass, right? And they gained mass because they had lost water in the hypertonic environment that we set them up in. We dissolved the shell, then we moved them into super sugary water, which was hypertonic. Then any fluid in the egg left the egg because it went from its hypotonic self to the hypertonic sugar. Then you pulled them out of the sugary medium and you put them in distilled water, right? 100 mils of distilled water. And that distilled water was hypotonic. The eggs were now hypertonic. Water must flow from the hypo, and it went into your eggs, and your eggs increased in mass steadily. That was all osmosis. Okay? And then that did not require any energy because water is just going from a higher concentration to a lower concentration. If you use a channel to do that, there are aquaporins for water, right? If you use a channel to do that, it's still not costing you any energy. If you have a gate that opens up in your cell membrane, a protein gate, that allows sodium to move from a higher concentration to a lower concentration, that's called facilitated diffusion. It's not costing you anything. A protein is just facilitating the process of crossing the phospholipid. Why do you need it there to facilitate it? Because sodium, that I just explained, it's a sodium ion, and an ion has a charge. And so it can't get through the barrier, that hydrophobic region. It needs a gateway to get through it, and it uses a protein to do that. However, if I'm concentrating sodium outside, and it's costing me energy to get it out there, it's called active transport, and that's usually called, starts with a P.
Oh, so potassium.
Sodium potassium pump. Yeah, the word I'm looking for there is pump. Okay, so if you go against the gradient, like you would in a sodium potassium pump, that's active transport, and that requires energy. Taking a whole membrane and engulfing something like endocytosis or spitting something out, exocytosis, you're moving all of your cytoskeleton. That's gonna cost you energy to move your cytoskeleton. Just having things ooze through your phospholipids, no energy. You feeling better now? Okay. All right. So remember we talked about, do you want me to take a picture of this too? Okay. Do you remember when we talked about what pickles used to be before they were pickles?
Cucumbers.
They were cucumbers. Okay, so cucumbers are bigger than pickles, yes?
Yeah.
So what would we need to do to make a cucumber turn into a pickle? Put it in a hypertonic environment. And if we put it in a sweet hypertonic environment, that would be a sweet pickle, right? And what's the other kind of pickle? Dill pickle, like if we put it in salt, right? Okay, if I wanted your red blood cells to start bursting left and right, okay, which would be death, and that would be bad, what would I need to shoot into your veins? It sounds horrible on this recording. What do I want to shoot into your veins? Distilled water. Because distilled water would be hypotonic, your red blood cells would be hypertonic. Okay, would plant cells burst?
No.
Why?
Cell wall.
Cell wall pushes back. That's called Turgor pressure. Okay, if I wanted your red blood cells to shrivel up, then I would insert what into you?
Hypertonic.
Yeah, super salty. Okay, and isotonic is like a 0.9%, but if I put a 9.0% in you, that'd be super hypertonic, and you're so, and they wouldn't function anymore. All right. So knowing that, okay. So we know the structures, right? We reviewed that. We talked about channels. We talked about carriers. We talked about going against the gradient with the sodium-potassium pump. Cell recognition. What is that? A glycolipid or a glycoprotein? Glycoprotein. Why? It's attached to a protein. And we talked about different diseases associated with each of these, yes? And then enzymatic, where it facilitates a reaction to take place. And then we said cell junctions. Who can't cross? Things that are larger charged, so you're going to have to use a protein to do that. And in the case of our lab that we did, could iodine cross our model of a cell membrane when we use dialysis tubing? Could iodine cross?
Yeah.
Yes, because iodine, which was in your beaker, went into your dialysis tubing and made the starch turn blue. Could the starch move? No, it was too big. And so your beaker didn't turn a blue color because it had to stay inside. And your sugar does move. Okay? So we talked about passive transport going with the gradient, either through the phospholipid or using a channel or carrier. If we use a channel or carrier, it's called what kind of diffusion? Facilitated. If we go against the gradient, it's called active, okay, like a sodium-potassium pump. Talked about aquaporins. We talked about diffusion and hyper and hypotonic. Are you all right with that? I'll review that. We said what would happen to the different cells in different scenarios. And again, there are pictures in your book where you can review that, as well as the questions at the back of your book. And then we talked about endo and exocytosis. I think we hit everything. You all right? Okay, do the questions at the back of your chapter so you're prepared for those. And for sure are there additional objectives or maybe not on this one? Are there? Okay, then do your additional objectives as well. And don't stay up too late. And if you're hungry, go get a piece of toast, but go to bed soon. Okay, love you very much. Hi, this is Miss Litton. This is still Honors Bio Review. Say hi.
Hi.
And we're going to do, this is still chapter 4 about membranes. And we've already reviewed that in an earlier video, but this is just all the questions. So let me get to one. And we just did anonymous, which is, oh. one anonymous student we're still waiting for. We're done waiting for you, anonymous. Okay.
Oh my.
Yeah, so carrier proteins are what? Specific. All proteins are specific. There's more of you than 10. Get involved. Yay!
Yeah.
Okay.
We're at 100%.
We are. Hang on to that thought. Two hundies. Hang on to that thought. Anybody need help with any of these? So just tell me if you do, okay? You're important.
Yeah.
3. Yay! Imagine it in your head, okay? So okay, that's all I'm waiting for. Oh. Yeah. So this one, a bunch of you picked C. Okay, when a cell is placed in a hypertonic solution, so here's a beaker, here's the solution, it's hypertonic, right? And I'm putting a cell inside of there. That means the cell is hypotonic. If it's hypo and the water is hyper, water must flow from the hypo. Got that? Okay, then if you want these other questions, just so we'll just do them out loud, okay? This is what? Exo or endocytosis or diffusion? Exocytosis, because it's what? Exuding. Okay? Which of these are methods of endocytosis? All of them. Pumps used in active transport are made of proteins. Pickles appear wrinkled and shriveled because they have been placed in a what?
Hypotonic.
High what? Hypertonic. Water must flow out of them so they shrivel down, right? And then I think that is the end. Okay, short little video. Be smart. 4 minutes. I can add that.